Hi, my name is Dennis Province from the IDEA National Resource for Quantitative Proteomics, and I'm going to cover in this short video offline fractionation and the purpose of why we do it and how we do it. So, many samples are too complex to run without fractionation. If you have a cell lysate, there are tens or hundreds of thousands of proteins in that sample, and when you break those up into peptides by doing some sort of digestion method like trypsin, which is our normal bottom-up workflow, we have too many things to analyze in too short a period of time. So the best way is to spread out the peptides so that we can analyze those and get better depth. So fractionation spreads out those peptides so each run contains a fewer number and depth can be improved. The fractionation takes time though. It takes time to fractionate the sample, and then you're going to have more runs on the LC-MS system. And so there's some sort of point of diminishing returns. So the goal is to fractionate the sample so that the peptides are spread out in a uniform way from sample to sample, so that we can compare them. And most TMT projects run in our core are fractionated into 46 parts and then recombined into 18 superfractions. So the fractionation that we have occurs in a 96 well plate. Well A1 and E1 are where the samples to be fractionated start. So we would fill those with our samples. The fractions are then deposited back into wells A3 through D12 for sample 1, and from E3 to H12 for sample 2. So usually we put 50 microliters of sample is injected, and we shoot for about 100 micrograms of peptides as the starting target mass. Our flow rate is about 75 microliters per minute, and the sample is resuspended in high pH buffer A. So our offline fractionator is not connected to our mass spectrometer, and we use pH buffers that are closer to pH 10 so that we can have a second dimension of separation, because our pH of our online LC system that feeds into our mass spectrometer is pH closer to 3. So we use a Chameleon software to do this, and we have to go into the instrument. We have to start the pump module. We have to turn on the UV light. So remember, the offline fractionator is not have a mass spectrometer connected. So the detector we use is ultraviolet light set at 214 nanometers. We have to turn on the pumps and the UV light and let it equilibrate. Now this is standard chromatography experience. Our pump module, again, we want to set it at 75 microliters per minute, but until the column temperature has reached its has warmed up, we start at 55 microliters per minute. We want to monitor the baseline. If we try to do that and the UV lamp is not ready, then it will tell us it's not so. Our column oven, we have to turn that on and wait for that to warm up. Now the reason why we have to start off with our flow rate initially set at 55 microliters per minute is because the pressure is really starts out at about 5,000 psi, and it'll increase to about 6,600 psi once the column temperature has reached its has equilibrated. And so the temperature will be too high if we start off with 75 microliters per minute, and that'll give us problems. So if you don't have this particular system, this isn't going to be very useful to you, but just so you know that we have to save our data, we have to add a new injection, we have to figure out what the method file that we're going to use. We call it a TMT Pro 46 frac. We usually just copy the file from the previous one, and we need to tell it what position that we're starting in. For instance, it says GA1, that's going to be the green. This is talking about which position of auto sampler that our plate is in, and then A1 would be where our sample is. And we start with an empty GA3. All the rest of the wells are empty except for GA1 if we're doing a single run. And then we hit save and wait. And so again, if fractionation occurs in the 96 well plate, the sample starts off in sample A1, and then the fractions for our particular sample collector actually come back out the same needle that the sample came in, and so it has to undergo some washing steps. It has to pass the sample through a column, and then as the sample elutes it back off the column, it comes back through the sample needle, and it will pass into starting in A3 and then go to A4 and then A5. And you might notice I've made a mistake here. It actually snakes around, so it gets to the end of A12, and then we'll go down to B12 and all the way back to B1, and then it'll snake from C1 all the way to C12, etc., etc. So usually 50 microliters of sample is injected and 100 micrograms of peptide is the target mass. So here's some typical traces that we see looking at the 214 nanometer trace. That's going to be the pink one for one of our sample sets here, and then the other one is the blue color. And so we have over the years developed a way of sort of being able to quantify the amount of peptides that we get. So all the way back in the protein BCA, when we tried to quantitate the proteins, when we started with our initial cell pellet and we made our lysate, we did a protein quant. That's where we started with the amount of protein. And then from that point, we went through reduction and alkylation, and we desalted and digested and desalted. And so there's quite a bit of loss at some of those steps. And so this is really the only other time that we can sort of quantify the amount of peptide that we're going to have. Now we also do, for our TMT experiments, we do a ratio check where we can quantify the amount of sample in each lane or in each for each label compared relative to each other. But this is a way that we can have an idea of how much peptide we actually have. And what we've developed over the years as the milli-AU, which stands for absorbance units, measured at 214 at the base of the peak that it has there, about the middle of the chromatogram, essentially represents the amount of micrograms that we would have. So for these two that we have here, so for instance for the sample set Higgs TMT, the concentration is about 60 or 65 micrograms. And for the van der Linden lysate, the concentration is closer to about 40 micrograms, just based upon these traces. And from that information, we can then make our superfractions at the appropriate concentration so that we can inject about 1 microgram per injection for each superfraction. So after fractionation, we need to combine those fractions that we have, those 46 fractions, into superfractions. And so we take that that plate that has those 46 on there, and we put it in the speed vac and we dry off the solvent. Two reasons why we dry off the solvent. One is because we want to get rid of the high pH solvent, and we want to add our low pH solvent to prepare it for the LC mass spec system. And the second reason is because we don't want as much solvent. So what we do is we actually put solvent in all of the wells in row B and half of the rows of C. So you can see on this diagram here the rows that are dark here in B1 through 12 and C1 through 6. Those are the 18 that we're going to put solvent back into. And then we're going to resuspend those peptides 6 at a time. So we're going to put a multi-channel pipetter. We're going to resuspend those pipettes up and down, and then we're going to take the liquid out of B1 through B6, and we're going to put it into 7 through 12, we're going to resuspend those peptides, and then we're going to put the liquid back into B1 through 6 again. Then we're going to remove 2 tips from B5 and B6, and we're going to transfer the liquid from B1 through 4 into A3 through A6, resuspend those peptides, and then put those back into B1 through 4, B4. And so what we've done there is we've combined some of the most hydrophobic peptides, which are going to be some of the more hydrophobic peptides, which would be D6 through D12, with some of the most hydrophilic peptides, which is going to be A3 through A6, with some peptides in the middle there, which is B1 through B6. And this is easy, there's lots of different ways that you could do this recombination, but this is an easy way to do it without messing up. Then you can see that would be for the green peptides. For the red peptides, you would take 6 more tips from a multi-channel pipetter, and you would take resuspend the peptides from B7 through 12, and then you'd take the C7 through 12, and then back again. And then finally, you would take C1 through 6, resuspend those peptides, gather the peptides from D1 through 6, resuspend the peptides also from A7 through 12, and put them all back into C1 through 6. So now, if you've done it correctly, you have taken 46 fractions and combined them into what we call 18 superfractions. And if you have run 2 samples, then the second part of your plate, which would be sample E, your sample would be an E1, and you would do the same thing with E3 through H12 as it's shown there in the color. So the calculations are, ideally we like to inject 1 microgram of peptide per superfraction. So if we have 18 superfractions coming from 46 regular fractions, and so if the initial sample contains 100 micrograms of peptide, then you would have about 5.6 micrograms in each superfraction, because if you take that 100 and you divide it into 18 parts, that's about 5.6. Okay, so if 100 microliters of buffer A, which is our aqueous solvent that's been acidified with formic acid, 0.1% formic acid, was used to resuspend the peptides, then each 20 microliter injection on the Eclipse would be 1.2 microgram of peptide. Did you get that? So if I use 100 microliters of buffer A to do that resuspension of those 18 superfractions, then I'm going to have 1.2 micrograms injected every time I inject in a 20 microliter sample loop. We want to ensure that there's at least 1 microgram of peptide injected per superfraction, and there's enough to make 2 injections. So we would actually put, we would want at least 45 microliters minimum transferred into our plate, so that, or used to suspend, so that if 1 injection messes up, we still have 1 more to go. I don't know if this is going to be helpful to you guys or not, but I hope it is.